First of all, I want to thank the organizers because this looks like a very nice, very interesting, exciting meeting and a wonderful setup. Let's see, I think just because partly of this, because it's a cozy atmosphere among friends and interesting people, I got inspired for trying to tell you something which I find quite exciting, but is work in progress, and so could be completely wrong. And I hope this is in the spirit of this. Okay, good. So I'm not responsible for what I'm going to say. So, among other things, therefore, you are guinea pigs, and I hope I will not make you suffer too much. So, as you probably know, or some of you know, we have been working for the last, my goodness, 12 years or so, on a boring model of the ventral stream, kind of trying to put in quantitative terms things that mostly physiologists have been believing as a kind of default model since Hubel and Wiese. And let me give you quickly some of the background here. These are images that you have seen already. I think Eero showed some of them. Ventral stream from V1, V2, up to probably different IT areas. In the macaque monkey, I guess it's about a total of 800 million neurons or so, 200 million of which are in V1, 200 million in V2, 50 or so in V4, rest in IT. I don't know, does that sound correct? And the usual story that has been establishing itself over the last, I would say, 50 years is that there is this hierarchy of different areas where you see an increase in the size of the receptive field. I'm speaking about fovea, then there's additional story about eccentricity, of course, that Eero was speaking about. But in the fovea, as you go up the ventral stream, you have this increase in receptive field size, and you have an increase in the complexity of the features of the optimal stimuli, and an increase in invariance to certain geometric transformations, like, for instance, position and scale. And these, of course, are cartoons, not to be taken too seriously, but about optimal stimuli in different areas, from segment-oriented lines in V1 to somewhat perhaps more complex in V2, and certainly more complex in V4, and so on. Okay, so the model we have been developing is essentially trying to be as much as possible faithful to what we know in terms of properties of cells in V1, V2, V3, and IT. So we have color here, kind of coding for V1 and V2, and layers of simple and complex cells. And this is, I want immediately to say, this is just one example of a broad families of, I don't know what the right name for this it would be, probably HW models, because the origin is Hubel and Wiese. At some point I tried to call them standard model, but I got misunderstood, because I use standard in the sense what physiologists mostly believe, and I think people took it as meaning something like the standard model in quantum physics or so. So anyway, so this is just to say that this version we have is probably one of the most detailed. It has millions of simulated neurons, but it's just an example of a broad class. And the first one really tried to quantify the ideas of Huber and Wieser was Fukushima. So he's really the first one to do this kind of things. And I don't want to get into the details of this, but there are basically two operations in the model. You can think of them as and or. You can think of them as one being responsible for selectivity and therefore for creating features of increasing complexity as you go up the layers, and the other one as something similar to an OR operation was probably something like a sum in the Huber and Wieser, going from simple to complex cells. In our implementation, is something more similar to a max among the inputs, but it could be a number of other things, max-like, sum of squares. So it's not to be taken too seriously in terms of the max. The tuning operation, the end, is something like a normalized dot product, which is actually turns out to be very similar to a Gaussian tuning. Tuning to an optimal stimulus that is the W there, is encoded in the synaptic weights. For instance, for V1, the simple cells, the W would be essentially oriented bars, or something similar like maybe oriented flanked by inhibitory surround. Okay, so and you go from simple to complex, getting some invariance to position and scale, which is by pooling over simple cells with the same selectivity in different positions and resolutions. And then there are more complex features at this level. In the present implementation of the model, these are learned by just random sampling of patches of natural images. So they're essentially the activity that a patch of natural image would elicit here, and this is coded in the synapses. So this is unsupervised. Until now, so the tuning of the S cells is done from unsupervised learning, apart very first layer in which we could do it, but for time reason we decided just to use Gabor filters, and then the pooling. So which ones of the simple cells are associated together in one complex cells in each layer? This is hardwired. Okay, so basically we have invariances to scale, but here you see position that are hardwired in the model, especially in the first two layers to translation. Okay, now a model like this seems to be consistent with a number of physiological data. Some of them, the blue ones, were in part used for setting parameters in the model. For instance, this old work we did with Nikos Logothetis about invariance in IT, to have some rough number of how many degrees of translation invariance there were for selective cells. And work with Jim DiCarlo. There is, for instance, this one with Ed Connor and Charles Cadio, in which I think we can account for the variance in the V4 cells for these curvatures patterns using a model like this. okay, so these are various comparisons, and they probably took most of the time of the last 10 years, including some psychophysics. This is a feed-forward model, so the psychophysics involves very fast presentations with masking, under the assumption that in that case you may not have top-down effects. So we are not dealing with attention. One cannot expect that a purely feed-forward model can explain all of vision. You need attention, eye movements, a lot of other things. Okay, they also, and this was for me quite surprising, like 5 years ago, made comparisons with computer vision system at the time on a variety of databases, and systems like this did surprisingly well in terms of detecting different objects. More recently, you have a system that, a similar system would be more a system of the dorsal stream that is start to be used by some of the mice labs in the Broad Institute and McGovern for quantitative phenotyping of mouse behavior by classifying, in this case, about 8 different micro behavior. You see the label underneath there, and this system performs more or less like the performance of a technician labeling the videos. And after that, you have 24 hours or more of statistics, and you can compute how much time is spent in each one of these behaviors, transition probabilities, and so on. And we can predict from the behavior alone different types of mutants, and then start correlating these quantitative measurements with genetic information, which ironically is much easier to get. Okay, but the point there is simply to say that models of this type seems to work reasonably well at the level of doing the task of recognition, so computation, and also being consistent with physiological data at different levels in different areas. Also, we did simulations at the level of biophysics, simulating neurons with realistic synapses and spike-producing mechanisms, so that we know these operations can be implemented with reasonable biophysical mechanisms. And so I think that's something to be taken into consideration. It's a lot of constraints if you have a model that passes test, or can be even just tested at different levels. It's too easy to make a model, say, of a visual illusion and forget what it's going to do in normal vision, for instance, or whether it makes sense from the point of view of being implemented with real neurons and be consistent with other data. So, however, for the last 10 years plus, I did not manage to understand why the model works as it does. What is about the hierarchy? Why there should be 5 layers or 10? What would be the optimal architecture, and under which conditions? And we tried to develop some mathematics, which is probably going to be useful, but really did not give an answer. And now, it seems a bit strange, but for the last few weeks, I think I understand. So that's what I want to tell you about. Let me start from this. The usual argument for hierarchical architecture of this type, from a point of view of computer vision or whatever, it's to create features of more and more complex, that capture more and more of the shape of objects, that essentially is like trying to create from an alphabet of lines words of more complex shapes, and then out of shapes, and so on. this has always bothered me a little bit. It seems, where does it end? How many layers should you have? And furthermore, recent papers, including Jan LeCun, and also some work we did, Joel Leibo and my group, seems to suggest that if you replace the S2 features that we learn from natural images with random features, basically the system still works. Okay? So features do not matter. What does? Okay. All right. So there are a few people involved here. More to come. You are welcome to help if this turns out to be interesting. So let me start from this question: where is the complexity of clutter-free object categorization? So the task here is distinguish a dog from a horse, and suppose and the difficulty. The difficulty of the task is that I would like to do something that probably a small child can do quite well. I would like to show you, or a small child, one dog, one example of a dog, one example of a horse, and ask you then to generalize to the other ones. And I would like to measure how good you are, right? And the question is whether the difficulty of this task comes from the variability within the class of dogs. We had here 150, I think, images of different dogs and 150 images of different horses, or it comes from the viewpoint variability. Each of these objects can induce, thousands of zillions of different images, distance, pose, illumination, and so on. So here, this is a database that was collected by a student of Oliva, and Joel got it. And so this here, everything is normalized in terms of viewpoint and illumination. So here is only the intra-class variability which is left. So this is not a proof of anything. It's just kind of motivation for what I'm going to say. So the question is whether this problem, once you have discounted, suppose you have a machinery that can be invariant to viewpoint, then is the problem still very difficult or not? And it turns out it's very easy. So here, the red one is a very stupid near neighbors classifier on pixels. This is the number of training example down there. So it's, speaking about not only one, but a small number. And I would say it does pretty well.
Can you just compare pixels, given that the dogs have different colors and shading?
Yeah, just pixel values. Yeah. Okay. So as you can imagine, I'll speak about, under the assumption that the main problem for biological object recognition is viewpoint being invariant to translation, scale, pose, illumination. And I'll speak about two issues. It's essentially a theory of how a model like HMax works. So it's consistent with it, but it's an interpretation I never thought about. And it also says ways in which to extend it. It says also how evolution may have generated, and a couple of other things. So one thing, it may be a bit confusing also because this is kind of the first time I speak about it. I will speak about theoretical arguments for learning that happens during, say, development of an organism. And I'll speak about once you have learned and things are frozen, how you process images. So, and I have three parts. One is describing this simple module that can provide an invariant signature for an image or a patch of image. Can learn and provide this. And this would be essentially what now is the network of simple complex cells, one complex cells and the simple cells associated to it in the model, or what Arrow has, in his model, because it would be very simple. Okay. The motivation is a theorem that has been used quite a bit recently in sparse coding and so on, which basically says that if you have objects that are in high dimensions, like images, and if you are not interested in reconstructing them, a relatively small number of measurements can keep the similarity or dissimilarity relations between them, okay? One example that fits this, you need more than one measurement, you need quite a few because you need to distinguish them, but you can project on a lower dimensional space. And the theorem also essentially implies that it does not matter very much which measurements you are taking. Let me give you an example. This is an old technique that was used for OCR, probably in the 50s. You just choose a small set of random line. I have a free there that I do. And your measurements are intersections with the character you want to recognize. The first line has 2, the other one has 3, whatever. And so you create a vector. This is the signature for what you have. It turns out this signature is pretty much diagnostic for the character. You can do pretty well. Of course, you cannot reconstruct the character. And the important thing is that these lines can be random, but you have to use them, the same one, always, right? Okay, so instead of measurements of this type, of course, you could just do a measurement of similarity with some template. That's what HMax is doing, simple cells are doing. The templates are this W, the synaptic weights. This theorem suggests which templates you choose. It's not so important. Okay? I'm exaggerating a little bit, but okay. So now, this is the motivation. Let me see. Suppose this is during development. Suppose I see an object transforming, any object. Could be a baby looking at the face of the mother, it's moving, translating, it's just looking at a patch. And suppose you have a machinery that records first frame, second frame, third frame. Think of them as simple cells. Maybe branches in a dendritic tree of a complex cells, but maybe. So essentially you have the frames of a video of an object transform. You're just recording these frames. Okay, so suppose you create this in this way. You create, it's a movie. This is the first frame, second frame, third frame. These are also simple cells pulled or connecting to the same complex cells. This is another video that you may take at the same time, another patch of the same transformation. Okay, I call this a template book. This is all is needed to become invariant to a transformation. And I call signature of a new image. So this is during learning. So let me jump now to how I'm going to use this. This is stored, that will be fixed. But later, if I want to use at this stage, I have an image, say F. The signature of F, the equivalent of my intersections for the A, will be dot products of F with this tau. Okay, so a vector of tau. See, it's like measuring the similarity of this patch of image with number of different templates. For instance, in V1 could be, as we see, like bars of different orientations. Okay, so the signature is a measure of similarity, normalized dot product of an image or a patch of image with this. Okay. Now, what I can prove is that if you take the max, if you do this dot product of f with all the templates you have stored, so not only this column, but all the columns, and for each line you take a max, that vector, that signature, will be independent of the transformation, in the sense that the signature of, let me see if I have it here, yeah, the signature of f and the signature of a transformation of f will be the same. There are some assumptions about the transformation being a group. For instance, let's assume that for now that this is translation. Okay. So that's pretty nice. It's a very simple memory-based module that just records anything that translates. And then after that, it can look at something, look at the signature, so that, and then if it looks at it again, this time in a different position, it will have the same signature. So it will be invariant too. Yeah.
Doesn't it imply, though, that the complex cell needs to be receiving afferents from the simple cells with delays? And is there any evidence for that? You need to, right? Aren't these templates coming in at different time points?
Well, let's see. So the learning during development, so the acquisition of the template, of this template book, this is done from a video.
Right, so that is over time.
Yes, but afterwards, when you're going to use this machinery, it's just a single image.
It's been collapsed.
Somehow. No, it's there. What happens is, okay. So here, this is the first frame, is the baby looking at the face of the mother moving. So this is first frame, second frame, third frame, right? It's just recorded. So for that, you need a video. You need to have a patch that transforms, translates. But then later, you are shown an image, never seen before. The rules is you do the dot product with all these things. You take a max for each line. You get a vector. That's the signature of this image. You put it in memory. And then if you, a second time, you are shown an image which is the same but translated. You do the same computation, and you will find the same signature. So you say it's the same thing. It's like a horse, same horse, but now, say, rotated. Signature, you compute it once, and so the interesting thing, you don't need to see that object in different poses or positions. You inherit the invariance from one template you have seen transforming. You inherit for every object.
Is this rigid objects?
Sorry?
Are these rigid objects?
Yes. What I'm considering now is even more than rigid. It will not be a big, we'll see, a big constraint. But what I'm going to speak about this, the transformation I'm speaking are affine transformations on the plane. This means translations, rotations, expansions, also asymmetric stretching. Yeah, that's it. This is the general linear group augmented of the translations.
Just a small question. I was wondering now, I'm a bit confused about how that is represented at the neuronal level. So
at the neural level?
Yeah, or at least, if you would think about maybe first time you see it, there is activation in simple cells, and then it converts
Well, let's go back.
Yeah.
No, because this, I'm not at that level. This is really, it's a good question, it's important, but this is kind of, at this point in an abstract machinery. I told you a rule to do it, and then I'll try to match it to how simple cells and complex cells could work. And as you will see, it's not everything completely clear. So you're welcome to provide ideas. Anyway, that's okay. So this is part one. This is kind of memory-based machinery for learning transformations and using them for guessing invariants. Yes?
So just, and maybe this is a little bit of a digression, but how integral is motion to this? And you're talking about the ventral system and motion-based learning as feeding in, or is the learning based on the fact that you have multiple images in a motion signal? And is that more the key element here?
Well, the idea is that in order to be invariant to a transformation, you need to record an example of that transformation.
That transformation is due to the motion, right? Or
yeah, that's through motion, right? I don't think there is any other way
to do it.
Yeah.
Okay.
I mean, affine transformations are motions in the plane. And, what would really be interested in is Euclidean motion, the perspective projection of Euclidean motions in 3D. But anyway. So now the question is, why should evolution have chosen hierarchies? In what I said, you could imagine that you have this module, and in order to be invariant to all affine transformations, what you'd need is this module to look at some patch of image undergoing all possible affine transformations. So it should record frames for transformation that are translation and rotation, and all combinations, for each translation, all possible rotation for each rotation. So it would be a very large number. I made some estimates, and you get, with some hypotheses about resolution and so on, you get easily to 10 to the 10 or 10 to the 9. So to do it in one layer, it's possible, but it's also motions of a whole image are not affine. Locally they may be. So there are a number of. But here's the interesting thing, which seems a little bit magic, is that transformation I'm speaking about are affine transformation, like using computer graphics. So this is a 3x3 matrix. It acts on vectors representing points, so x, y, and 1. This is just to give the ability to represent in this way also translation. So basically these 4 numbers represents rotations, expansions, stretches, and this represents translation in x and y. If you operate on a vector, they do the right thing. Okay. Now it turns out that this is a group, which has an inverse, is not commutative, and so on. It's the affine group. There is a certain mathematical structure, and one of the properties is that you can factorize this group in this way, like this part that represent rotations and expansions, and this part that represents translations. Okay? So now the idea is, if evolution could say, okay, I will evolve V1 by showing only translation. So V1 learns only translation. Then you have already simplified the problem quite a lot, because instead of having to store, like in the one layer case, say n time n2 transformation, right? Say n1 translation and n2 the rest, you can now do n1 plus n2, because you do it sequentially. So the idea here is that because of this factorization, you could save a lot of storage, go from 10 to the 10 to 10 to the 4 or so. And now is the other insight here. We don't have a proof. I don't have a proof clean, but it seems pretty obvious that. Let's see that. Okay, that if I look at some pattern which is translating or moving, say moving, and I look at it through a small aperture, the only thing I see is translation, even if it's rotating, unless I'm really unlucky. So the idea is that if you look at the world through an aperture is small enough, and small enough is related to the resolution of the image, say neural image you get from the LGN, then you see translation. So here evolution has a possibility. If you impose these small receptive fields in V1, it's like training V1 only with translation, because that's the only thing that each aperture is going to see. And so that's the idea for having a hierarchical architecture made up of small and then larger and larger apertures or receptive fields. And so the idea is that you'd have this from these properties of the group structure of this affine transformation, you'd expect to have, for a small receptive field, to see, estimate, learn, be invariant to small translations, and then rotations and expansions, and all of this would be true for any type of objects. And then higher up, you can expect that the same memory-based module could learn in this memory-based way, store examples of transformation that are not affine and may be specific for a class of objects. Like, for instance, if I change expression in my face, that's something very specific for faces. If I collect examples from one face of this transformation, I can use this to be invariant to that for other faces, but not other objects. Rotation in depth of a face is another example. There is no way I can learn generically for any object from the image alone, this kind of transformations. But if I can learn a good approximation using that memory-based module. And we have done that. So, for instance, this is an example in which Joel Leibo and Jim Match have used the usual HMax, but added a layer at the top in which the templates have been learned from a face rotating. Okay, one face, and then the experiment is you show to the model one face, say frontal, of a specific individual, and then you try to recognize whether it is the same face or not when it's rotated by 80, 100 degrees. So you have never seen that face rotated. And if you do that with a standard model, you get an accuracy which is pretty good when you look at the same face rotated only a little bit, 10 degrees, but it gets quickly pretty bad. If you do it with this module that has seen another face rotating, you have not perfect, but much better recognition. Is that clear? So this is the third part, and this has to do with trying to connect everything I said now with properties of cells and tuning of receptive fields. This is a bit more speculative. It's mostly addressing what happened, say, at development or during plasticity. So the idea is to use some Foldiak type rules. So Foldiak suggested the way you get wiring of simple to complex cells was essentially temporal correlations. If you have simple cells that fire one after the other, they are likely, assuming that the simple cells are already there, have their specificity, if they do that, it's likely there is some stimulus that is optimal for one and it's optimal for the other. So all of them represent the same thing. They can be associated together. It's like the invariance to position here. Okay. So the idea is doing something like that. Keep that in mind. So the pipeline we have is we have images. We assume that we have a retina doing very roughly something like temporal derivatives and Laplacian over Gaussian, so spatial derivatives. And then we have V1. But let's look at what happens at the output of the retina. Okay, so if I take images of a natural image, this picture, translating, and these are taking frames, and I build my template book. So and then I look at the spectral properties, say principal components, actually, SVD of that tensor. What I find is this. These are. This is the first 4 principal components, assuming an aperture receptive field size of this, and here is larger. So they look like Gabor frames. The reason intuitively is that you have, the eigenfunctions of the translation operator on R2 are Fourier functions. Here I have, we can get more in detail, but I don't have much time. Anyway, now this was for vertical motion. This is for horizontal motion. So you find the orthogonal ones. This is a section you find in quadrature. The first 2 eigenvectors. This is doing the same thing with noise pattern instead of natural images. So this does not depend on the statistics of natural images. Depends only on the transformation. I'm saying a lot of, I guess, provocative things. This is what you find for rotations. So same thing, as I said, the aperture are small. Rotations and translations are the same. And we are starting to look at principal components in not in V4, at the level of S2. We find things like this. This for rotation, this is actually what you predict, because the theory says, see if I had this, maybe I don't, but the theory says that we should find essentially features that are orthogonal to the trajectory of the Lie group, of the Lie algebra that generates the affine group. Anyway, we can discuss later, but hopefully we'll find things that may remind some of this. That's all. That's, I hope. Why do I care about the spectrum and why do I relate it to receptive fields like that? Because here's another assumption, is that Hebb type rules, there is in particular one by Hoya, do extract principal components from the inputs. And so if you have Hebbian type rules working at the synapses of simple cells, and if those are the principal components of the template books, you could expect during development, because the system probably does not want, could, but does not to keep all those just straight raw recordings of movies, wants to compress them. The compression will be principal components done by this quite natural Hebbian rule. Okay, so let me try to put things together a little bit. There are a lot of holes, and I apologize, but so there is this basic memory-based module that could be used, and yeah, I'm not sure exactly. I think each simple cell, like in, would be very similar to what we have in the HMax, only that there it's hardwired. It would be, frames. So, for instance, of a bar moving, translating, and all of those will get wired to the same complex cells. And that's a basic thing. But then you have this idea that the reason for hierarchies is in order to optimize the storage, minimize the storage requirements. And so evolution should have chosen this hierarchy in order to essentially automatically present only translations to the first layer by using small apertures. And by doing this, you have different transformations learned in different areas. The different transformations reflects themselves in different principal component spectrum of this, what each layer stores in the template books, and then if there are Hebb-like synapses, you get these spectral properties reflected in the tuning of the cells in that area. Oh yeah, that was what you expect. You expect features that are, for instance, here, this would be the kind of features you'd expect for expansion. And these are the ones for rotation, orthogonal to the motion. There is this prediction of class-specific areas or patches. The fact that, how strongly neurons respond is not so important. Invariance is more important. Statistics of images is not important. Yeah, there may be something about whether simple cells are just failed complex cells that have stored only one eigenvector. Anyway, sorry, I probably went over time. Thanks a lot. So I know it's a lot to digest, apologize, but I'm quite excited because that model we worked on, I always thought at the beginning was something we were going to, 10 years ago, work on so it does not really work for recognition, leave it and go to something more interesting. But I got stuck with it. I found it always a bit boring. And now it's kind of ironic that the theory that is mostly consistent with it is actually quite exciting, at least for a physicist, group theory. Anyway.
I have a quick question. So the rule you're describing is a rule for how a complex cell should pool over simple cells, right?
And how simple cells should
generate their responses from their
yeah,
afferents?
so it's
so
mostly
that's
a
the piece
rule.
I'm not seeing.
Is a rule doing both things, possibly at the same time, maybe with different time scales. So the main important one is the connection to the same. So you have LGN, suppose, and when they are their activity close in time, you connect these things to the same cell, say complex cell. And you suppose that the synapses, at least in this naive description, the synapses of the LGN reflect exactly the frame, the image that they, the pixel that they have seen after this filtering.
Yeah, but you need something more than that for later areas, right? The simple cells, you have to describe the structure that's driving those. And
Yeah, but
seeing where
later
that's
areas,
coming
if you have the output of the complex cells
from the previous.
previous, those would be like an image, right?
Mm-hmm.
I did not show some of the results, but the invariance of this signature behaves surprisingly in the right way. So,
but something has to kind of break the symmetry and decide which things to represent, and that different cells should represent different things. Especially when you have, for example, an area like V1 that projects to many other areas, and each of those other areas does something different, right? So the simple cells in those other areas have to learn different kinds of features that are relevant for their computation. So it's not clear how you break that symmetry and how you decide what those things should be.
So I mean, it's the usual story. Let's see. I'm speaking about, view invariance important for recognition. There are certainly things that you want to do that require the opposite, require exact localization. And
like when you go up the dorsal stream.
You may do that, right? Or there may be even in the ventral stream enough information left by. Maybe that's what the simple cells are doing. Yeah.
Failed complex.
I don't know. I would be curious to know if you think there are some obvious flaws, before I waste too much time in the next few months. It would be useful to know. Yes.
This is more on the philosophical side. So, if you store signatures of different transformations at different levels, if I understand, different areas. So this would be, sorry? Sounds like, to me, like just the opposite of grandmother cells, in the sense that to decode an image, the meaning of an image, you actually need to entertain all the signatures at all different levels at the same time, right? Would that be?
Well, let's see. In the current HMax model, the equivalent of the final signature is about, I forgot the number, by a few hundred C2 cells. So it's a vector of, few hundred components, which is the output of the last stage, which is the receptive field there, just in the model, is covering about 4 degrees of visual angle. So, the fovea, say. And that's the signature. So that's the only thing you need to pass to other stages, like a classifier. Or I prefer to think in terms of associative memories. So that's a signature of an image, which is invariant to various transformations, and which you can use to retrieve memories associated with it. So if you have an associative memory, which is a kind of old version of a classifier. You can store two items, two vectors, say, A and B, and then by presenting A, you get out B, and you can store many of these pairs superimposed and still have.
But these vectors are across the hierarchy, right? I mean,
No, in the HMax, so you could get signature at each layers, but and maybe that's what top-down signals do, science fiction. But for the reason how we use HMax now, we get only the final signature, this vector of 100 numbers at the end.
So I was just wondering how to exactly relate what you said about the transformations, that they are very important to this idea that natural image statistics sort of shape the tuning of different cells, because now it seems to be a bit at odds with each other.
Yeah, I think it is at odds.
How to relate them to each other? I don't know if you have any.
So the extreme version is, that's wrong. And receptive fields are really shaped by transformations. Maybe, I showed you we can get those receptive fields from random noise. What is important is the direction of motion, the type of motion. So this is the extreme answer. There is something that I did not mention so far because I don't know anything about it. But so what you'd like to have from what I call signatures, some representation is invariance, that I think is important. But also, of course, you want also discriminability. So you want different objects with different signatures, right? And so there is this part. And so this may give back some importance to, statistics of natural images.
So I was just sitting here thinking about what the physiological predictions are. So does this specific instance of the general idea predict rotation invariance in area V4, for example?
Yes, but it's so what I don't know. So I don't know. Let's see. How should I say? From the theory point of view, the rotation, expansion, and stretches are transformations that commute. So I cannot use the argument that I use to say translation first and the rest after. The rest can be factorized in different ways and does not need to be factorized. So we are doing simulation to try to sort out, understand what would be the best way to do it if there is. So it could be a mix, but the prediction would be mostly a mix of rotations and scaling or stretches. Personally, I don't think that in terms of visual experience, you would have many motion transformations that are really rotations on the plane. Mm-hmm. So I don't think rotations are the more important one. I think expansions and stretches are more important.
Okay. And another question about physiological prediction. So does this predict responsiveness to static, radial, and spiral patterns of the sort that Jack's work describes?
Yeah. Yes. Those would be, these are the ones that I cannot show you. These would be examples. These are PCAs, SVD, at the output of, in the standard usual model. This is what happens for the C1 level. there are C1 of 4 different orientations. These are the principal components. Let's see, the first 4 principal components for each one of these classes of C1 when there is a natural image rotating. So I'm not sure what exactly would you know, a cell that will combine this, because we are thinking of the S2 cells, this would be like V4, combining all of this. So you may see, here I just superimposed them just an hour ago with different, these 4 here. I would expect some structure in this case that is like, you know, like this, for instance, which you see kind of, because this corresponds to features orthogonal to rotations. I would expect things like spirals, and so because of the combination of expansions and rotations. It's essentially, features would be, you know, roughly speaking, orthogonal to the trajectory of a linear differential equation in 2 dimensions, which are, you know, there is a very good classifications in terms of what can happen there. Those are the affine transformations. I would also expect probably some translation to be left, because the small translations will be eliminated essentially by the V1 stage. But larger translations are still to be present there. So, so I think will be more of the mishmash, not as clear as in C1, but yeah.
Tommy, I had one question about, related to Eero's question about the selectivity, and maybe that this model is lacking something in terms of determining what those S-like units should look like. And you should talk more details about that algorithm and those receptive fields that you showed up there. But it's my intuition that without some sort of like aperturing like effect with the algorithm, like an aperture or a windowing like effect, you actually won't get those like spatially selective receptive fields that you showed there.
Yeah.
So for example, a very simple selectivity just in terms of space, just in terms of the spatial location of like a simple cell, it seems you need an extra ingredient to this algorithm in order to get that. And I assume that you're aperturing or windowing those.
Well, the aperture is really, let's see, would be biologically correspond to, you know, how large the dendritic tree is in terms of visual angle. That is a natural way to provide an aperture. Is not
Right, certainly, but it's probably also we should look at the numbers, but they might actually work out such that the receptive fields could be much larger than the ones you actually find. And so, in fact, you have, through whatever developmental mechanism or learning mechanism, pruning up of these S units such that they are quite selective in terms of spatial position. And that then, implies some sort of additional learning algorithm in order to become selective in that way.
I don't think so.
Okay. Right, and I guess then that is the mechanism, though, and that seems like it has a little bit to be desired.
Well, that's kind of a hypothesis, right? Because Charles, you're saying there has to be a learning rule to learn the specific thing, and Tommy's kind of implying that it just has to do with the receptive field size, that basically you get it for free. But it seems like you could look at that by looking at the statistics of natural movies and trying to determine if the statistical presence of affinable transformable image patches basically varies with image patch size. At some point, when you get down to really tiny sizes, of course, as you said, everything turns out to be translation, where tiny size is defined in terms of the sampling grid.
Yep.
But the question is, how do things change as you go up? I'm
so
very
but then for very low spatial frequencies, then there's a little bit of a
Sorry?
conundrum here. So for very
Right.
low spatial frequencies, there's a little bit of a conundrum where you have very large receptive fields for low spatial frequencies, or
Yeah.
larger receptive fields. But then this theory should imply that the arborization and the selectivity should be the same, though, for those low spatial frequencies.
Does anybody know what is this situation in terms knowing from the LGN the size? You know, are there, say in the fovea, more than one size of center surround ganglion cells? There should be, right? But yeah, but you know, the old story was you have already in the, say in the retina, in the fovea, different, for instance, in the cat, you know, X, Y, W. The X, Y would be, what was? The alpha, beta would be like the magnum and whatever it is, parvo, yeah. So at least two sizes, but probably more, because it's related to this story, right? We're assuming this center surround filters coming from the retina, and what would be nice if you have already different sizes there. They will determine a match between essentially the receptive field size and the bandwidth.
Well, I think the retina guys currently say that there are about 20 different cell types, right?
Yeah.
And each one of those types has a particular size. They're different, and each subtype tiles the surface of the retina. They all probably have eccentricity dependent scaling of some type.
Yeah.
They often come in both on and off varieties, but not always.
Right. Yeah. But you know, it may be like in the cat, the story was there was a X, Y, alpha, beta, and then there was the W or gamma, and W or gamma was everything that was not going to cortex, like direction selective cells, and you know. Anyway, I don't know what the story is with the monkey.
Well, I think Charles's request to have a learning rule that learns this is valid. It's me back here, because if you take the principal components of natural images and window them, they'll end up, you know, the first order, the lower order principal components look like Gabor functions, and the higher order principal components look like Haar wavelets. And then when you window them, they end up having some sort of circular symmetry. So I think it's valid to sort of want to an existence proof in an artificial system that you can actually learn rotational invariance. And I mean, just so you know, I'm very sympathetic to this. I don't know if you remember, but 20 years ago when I first started doing that non-Cartesian stuff, it was to test the Lie group theory of vision. So the whole point was to look for rotations and dilations and stuff. And
yeah, no, you're right. Yeah.
Yeah. So, you know, if you kind of look at the history of physiology since that time, there's not. I mean, there's evidence for size invariance, which you can think of as dilation, and there's evidence for some sort of shear. Shear is the other property you didn't talk about, right? Mm-hmm. The fourth group is shear. There's evidence that cells are invariant to shear. That's okay, good. There's not a lot of evidence in the ventral pathway for rotation invariance, but in the dorsal pathway, you get an MST, you get rotation sensitivity and dilation sensitivity, cells that are tuned for those. So it's kind of a complicated, ugly system, as vision always is, with stuff that you'd want to be together, kind of spread out all over the place. And it's kind of unclear how you're going to make a learning rule that's going to be selective for, you know, just these properties. And for the example in the case of V4, what you would like to see is that if you had a really dumb learning rule that only had big receptive fields with long temporal integration, that you could end up getting something that was selective for polar patterns, for example. That would be really cool if you could do it. And I haven't seen any evidence that you can yet. No, in V4, though, V4 has a very long temporal integration window. So for example, you could imagine setting up a situation where you have large receptive fields with short integration, and then you'd end up with MST. And that should be sensitive to rotational motion. And then you'd end up with a large receptive field with long integration, and that should be selective for concentric patterns.
Well, that may be related, you know, to translation can be fast, microsaccades and so, but looming is slow because we have to move, right?
Well, or if it's fast, you have to get out of the way really fast.
Sorry?
Or if it's fast, you have to get out of the way.
Yes. But well, I don't know, if we raise a kitten in, like Colin Blakemore did a long time ago, but this time in a noise environment, random dots environment. So the prediction here is that it would be fine, because transformations are the important thing. And one could do the experiment in V1.
That will be the psychology equivalent of Schrodinger's cat, Poggio's cat.